Trevor gave me some prompts before that really go outside of my slide universe. My slides already take for granted that everybody knows what I've done before, and they go essentially beyond it. So essentially, to pick up from what Trevor mentioned, I found myself in a very strange situation when I started, because I started in Munich, and I was with my mentor Walter Hoppe, who was an X-ray crystallographer. He was, in fact, briefly at the MRC, and he developed this idea of getting into electron microscopy. So here he was with a tremendous theoretical background in Fourier theory and the X-ray background, and he had this idea of doing electron microscopy, which really, supplies the phases for gratis, and you don't have to work for them. So I was supposed to do a 3D reconstruction right there from scratch, and so he had the idea that just by changing the programs a little bit, then I could get this done. And then he just didn't have any idea about the properties of electron microscopic images, which are so terrible, and so they require really a study on how, the properties. But at the same time, the 3D reconstruction was already done in Cambridge. The work by de Rozier and Klug, 1968. The first application of the computer in doing a 3D reconstruction using a method that had been developed by John Radon in 1915 or so, at the time when no computers existed. So I found myself in this very strange position, because the work had already been done. On the other hand, there are severe limitations, because what de Rozier and Klug worked on were the bacteriophage tail, which had very high symmetry. So molecules were essentially crystallized into a compact form in very rigid spaces. So it was a situation in which programs had to be developed specifically. They were tailored to a particular symmetry. And the same thing happened with the spherical viruses and also planar crystals. So you always had a situation of specifically tailored programs. And I was interested, and my mentor was interested in solving the problem in a completely general way. And that Hoppe had the idea of putting the molecules in solution onto the grid, of course negatively stained, because no cryo was available, and then essentially doing electron tomography of these, so you reconstruct the entire grid along with the molecules sitting on them, and then of course you get a very noisy representation of each molecule, and then you can take them and maybe orient them with one another in order to get an three-dimensional average. So that was the approach, and I thought it was deeply flawed, because you would overexpose a molecule as you do the 3D reconstruction, as you do the data collection. And that's how I got the idea of the single particle method, where I thought we don't even have to tilt the molecules. They're already tilted. They have already tilted themselves in solution. They are in all different orientations. And that's how the whole idea developed. This is the single particle method. And when I started this, everybody thought it was completely stupid and it would never work and so forth. And just by being persistent, I got it to work. However, at the very start, with that kind of concept, you have only solved a very small part of the problem by doing the data collection, because then you don't know how the molecules are actually oriented. So all the tasks of data analysis had to be solved after the fact. After you don't know the projection angles, you don't know which molecules belong to which, and so forth. So all this had to be solved by programs, and my claim to fame is really in the developing these programs and using math and computation to get to a point of 3D construction. Okay, so that's how the whole thing started. Okay, now I get into my my contribution, which starts from the idea that you already know all this, and in fact, the contributors to this workshop have already implemented very sophisticated versions of this idea and are working on the forefront of this technology. And I would just wanted to talk about the particular a particular property of molecules that make the of drugs possible. And you cannot understand it by just looking at the static structure. You have to look at the way the molecule actually breathes and moves in the thermal environment. And so the very important aspect is really the aspect of conformational changes. How do we get information about conformational changes? So we know that small drug binding at a target site can be facilitated by allosteric effects of a ligand that is far away, that is very often used now in the development of drugs. And that, to me, is reminiscent of the Hirsch suppressor mutant, the tRNA tryptophan harboring G24A mutation, which is far away from the anticodon and allows read through. This can only be understood by the idea that this mutation changes the conformational spectrum of tRNA. So allosteric modulators are those that the binding of ligand modulates and strengthens or weakens the binding affinity to a ligand that is in place. So my contribution now is focused on the methodology on how do we get information on the conformational spectrum of a molecule. I have not, except by in some collaboration, I have not made any contributions to drug development myself, but rather I thought talking about this methodology would in some way advance the issue. So there are two new techniques for gaining understanding about the dynamics of biomolecules. One is time-resolved cryo-EM, which means that we go from the pre-equilibrium situation to equilibrium. We follow a reaction actually in real time. And the other one is manifold EM, or other techniques for resolving heterogeneity applied to images of molecular ensemble in equilibrium. That is we do an immensely large data collection. So we get a very large ensemble of molecules that assume all states that are compatible with the thermal energetic environment. Um, and then in order to always keeping in mind to get information about the dynamics, these this information coming from cryo-EM can be combined with two other biophysical techniques that have nothing to do with cryo-EM, namely single molecule FRET, where you can get information about distance changes in the molecule in real time. And the other one is HDX, the hydrogen deuterium exchange combined with mass spectroscopy. So there you can get information on sort of binding opportunities that come along as the molecule breathes and goes through its conformational spectrum. And I'm focusing, of course, on 1 and 2, because that's where I was able to make some contributions. So here, this goes back to a device that was developed while I was still at the Wadsworth Center in Albany. There was a collaboration together with people at RPI. And the simple idea is that you have a microfluidic chip. We have solution 1 and solution 2. You mix them in a mixer, and this is a 0.5 millisecond only. And then you have a reaction channels. We have different lengths depending on how long you want the two already mixed molecules to react. And then you have a gas assisted spray onto a dry grid, and as the grid falls down, it gets sprayed and covered with droplets, and goes into the cryogen. So for different time points, you have different chips with different reaction channels. Now there are other methods of time resolved cryo-EM that have been developed, where the mixing occurs on the grid itself. And so this is a sharply different method, because you want to have control over the mixing and the reacting. If you apply it on the grid, you essentially don't have control. Something is happening on the grid. You don't know what happens out there in the air-water interface. There's no sharp division between mixing and reacting. Okay, so that's why we focused on this one here. So the history of this, the first experiments were done at the Wadsworth Center, and then I transferred the whole technology to Columbia. And this is sort of my hero here, Bo Chen, who really was able to put it all together. And he made a collaboration with the engineering department. We were able to transfer this all. And I particularly have to mention Howard White, because he has been doing time-resolved experiment in the muscle field for quite some time. But it's essentially with the other method of mixing on the grid. But we got an apparatus from him which drives the entire thing, the pumping and the plunging. So everything is done under computer control, and we still use his apparatus. And so this is really sort of the infrastructure is all computer controlled. You have an environmental chamber you have syringe pumps that pump under computer control, environmental chamber, the gas pumping system, and so forth. Mm. And this is where this is how the initial device looked. Um, this here is the environmental chamber, which is attached to the apparatus that I was telling you about. The chip is mounted horizontally right here. There's an inlet 1, inlet 2, and the sprayer is up here. These are is the gas that drives the sprayer. And then it sprays into this direction here. Here's the grid. The grid is mounted on the tweezers. The tweezers fall down into the dewar with liquid ethane. And so I'm just going to show you a study that was done with this device, and then I'm going to go on to what we developed afterwards. So we went and developed something that did not involve a silicon chip afterwards. So this is when you study ribosome, which I've done extensively, we are looking at all the kinds of different phases of this process: initiation, elongation, release or termination, and recycling. And you can take a pick anywhere and study intermediates that are not even shown here. So this is a vast territory for research. If you want to really dissect it and see what is happening in each of these steps. So here in a termination, we are looking at this particular process. So in which a stop codon is presented on the A side, and it sort of invites the binding of an release factor, which can be release factor 1 or 2. And so then the binding occurs, and the release factor makes a very large conformational change to reach the peptide transfer center, where it essentially clips off the peptide by hydrolysis. And the fascinating thing was that RF1 and RF2 are in a compact conformation as known from X-ray crystallography, whereas this conformation here is completely different. One of the domains is sort of stretched out, and so something happens at the time that the RF1 or RF2 binds to the mRNA, to the stop codon, and this change had never been seen before, and we had the ambition to visualize this in real time. And oops, this doesn't work. Okay, that's what I'm afraid of. The movies simply don't work if you yield control to. Okay, so what this supposed to show is the change in conformation of the of an release factor as we bind. So it expands. Okay. Yeah. Okay. Thank you. All right. So I really need cooperation now, because I have no control over this. So you see the change in conformation that had been shown before. Well, okay. So, and from the kinetics, in kinetic studies, we know about the existence of this of an intermediate state of RF1 in the compact form on the ribosome. So, which only happens for 20. It reaches the concentration reaches its maximum at 20 milliseconds, and then it rapidly falls, which simply means that with ordinary cryo-EM, you will never be able to catch this. You need a device like this to do it. This is just the same here in an expanded time scale. Okay, so we want to catch this particular conformation. Right. So this is what the experiment looks like. We have the termination complex, and then we have RF1 or RF2 in the compact form, and then at 24 milliseconds, which was the time for which this device was designed, we actually see the compact form of RF1 bound to the subcodon. Okay, this had never been seen before. Now we do another time point, 60 milliseconds, and we see it in the expanded form. Okay, so you can really see this is sort of a simple movie, a movie with only 2 frames, but it's a very important movie. It shows you biological results that have never been seen before. And so on account of this and other experiments in which we looked at other processes in the case of the ribosome, we believe that this is a very important technology that should really be put in the hands of every research group which is doing. So again, to summarize, here's the pre-accommodated state, which we were able to capture at 3.9 angstrom, the accommodated state at 3.4 angstroms. And now the new setup is right here. And it's well, it's very difficult to see here. And we have now we're using PDMS, which is a plastic, and it's much cheaper to use and much faster. It's much more versatile in our hands. And so it completely replaces the silicon. It's very difficult to see here, but this is the mixer right here. And then the reaction channel is very long in this case. So the entire thing is modular. We can sort of plug in with different mixers, different reaction channels, different sprayers. So here's the mixer, here's the sprayer. In between is a very long reaction channel, which is made out of glass, capillary. One important thing that needed to happen here is it needed to be coated from the inside with silicon oxide in order to prevent the adhesion of protein, and so that we had a very clean process, because otherwise we will get irreproducible results. Yeah, I noticed that my new PowerPoint was not loaded. It's the old one. So here, with the new device, which I wanted to show you a few more things, which I cannot do right now. So this is the experiment that we were able to do. And it shows, let me see what's in the next one. Is okay. All right. This is a situation in which a prematurely terminated ribosome is rescued by the action of an enzyme called HFLX. It's a GTPase, so it arrives together with GDP and lodges on the ribosome and recycles it. It splits the ribosome into its subunits and then recycles everything. So this is how the bacterium is managing a situation, an unexpected situation in which it prematurely terminates, for instance, by the action of a drug. So this is essentially a drug resistance problem that we see here. So the action of this small molecule accomplishes a splitting into the two subunits, and at the end, it stays with the large subunit. And so we wanted to know this particular process, and we find that we actually get three intermediate states. When you look closely, and I will show you in a moment, the ribosome opens stepwise like a clamshell. And so the process is very organized, and the small molecule HFLX is lodging in between and then expanding in a particular way to accomplish this feat of splitting the ribosomes. Okay, here we don't show the HFLX, but I can just show you the three steps, 1, 2, 3, and then in the end, we only see the 50S subunit. And you have to appreciate that the ribosome has at least 12 inter-subunit bridges, which all have to be split. And it turns out that the last two bridges that get split are B3 and B7A. So these two bridges act like a hinge of this entire clamshell process. And so I wasn't. It's very difficult to show both things together because it's a very small molecule, and I just show you what the conformational changes are that we see of HFLX in this process of the splitting. Okay, so that brought us to this place, and we are really convinced that for processes that are in the range of 10 to 10 milliseconds to 1 second, this is going to be a very important technology. And this kind of time range is important for molecular machines. Always, when you have a biological process in which a very large domain changes position, then you get this kind of time scale. And but we were unable to get into a range that is lower than 10 milliseconds for a variety of technical reasons, and one of the most important reason is that the plunging process itself takes a certain time, certain minimum time. So in order to get into another time range, which is important for receptors and ion channels and so forth, another kind of technology needs to be developed. And that's in progress in the engineering department. So I want to talk about something completely different, which is the capturing the conformational spectrum of molecules in equilibrium. And as I will show, it is a way of getting an estimation of the free energy landscape. And this here is pure fantasy. It's just it's supposed to make you excited about this. And so the idea is we want to infer molecular motion in a continuum from an analysis of a very large single particle data set. And that goes back to collaboration with Abbas Urmas and Peter Schwander at the University of Wisconsin at Milwaukee. Ali Dashti was a postdoc there. Peter Schwander was a graduate student in Abbas Urmas' place back in 2008 or so, and he is now a faculty member. So I'm still collaborating with him. And then later, when we got into applications of it and further development, we have here these people at Columbia University. Amédée is now an independent researcher in New York. Evan Seitz has been prolific in exploring these ideas and getting beyond it. And these are all the applications that came out of this, and they are the most recent is really these two papers, which go beyond the initial set of programs that was developed, and the most comprehensive publication that deals with, in particular, biological application is by Dashti et al. In 2020, retrieving functional pathways of biomolecules from single particle snapshots. And this was an application to the ryanodine receptor, and I will show you this in a moment. So the idea is to look at the idea is the following. We want to really have information about the conformational changes that the molecule undergoes in its thermal environment by looking at a very large data set. And how do we do this? Well, when we do normal data analysis of single particles, then we first do an orientational classification. And then we get essentially in a particular aperture that is characterized by a particular projection direction, we get sort of a subset of the data. And that subset we now analyze by looking at the manifolds. So the images can be thought of vectors in a high-dimensional space. The dimensionality is simply the number of image elements. And so we have the data, they are distributed in some way in this high-dimensional space. The conformational changes means that some trajectories in this high-dimensional space. And we want to characterize, we want to follow these trajectories. So we do this analysis for every single projection direction, and in the end, we make an consolidated mapping everything that we find. So the idea can be now imagined in the following way. There's a place in China where you can see 10,000 horses galloping along. You are in a particular place as a reporter or a journalist and look in this direction. You see each horse only from one particular direction. Now you can really for that direction, you can now order the horses according to uh, the uh, pose that they have in the galloping. Okay, so there's always for every single pose, there's always one that is before and one that is behind. So you can order all of them. Yeah. Once you do this, you can just make a movie out of this. Okay. You don't know whether a horse goes forward or backwards because we have no record of the timing. Okay. And this is just choosing the forward direction. But this gives you an idea. Now, how do you collect data uh, according that are ordered according to similarity? So and this is a one-dimensional problem because there's always, the galloping goes in one direction, but you can also have conformational changes that are two-dimensional or three-dimensional and so forth. Um, so So, so this goes back to something that I did a long time ago already. We discovered that one can order images according to their similarity, but Ba's group really made this into a very exact science. So we have an image, and which is a vector. The vector has the dimensionality of the number of elements, and um, if you have continuous changes in conformation, then it means that you're sort of going from a particular place in this high dimensional space to the vicinity in some trajectory. And no, the important thing that needs to happen is that we have to go from the high dimensional space into a low dimensional space by some technique of dimensionality reduction. We really look at the conformational maps in a space of 2-dimensional or 3 dimensions. So we essentially follow the direction of the changes in this high dimensional space, but we essentially flattening the way the cloud is curved. Okay, so now if we get a conformational mapping, then we know exactly for each pose. For each pose, we know exactly how many occurrences, how many sightings we have, and that's an important number because when we see a lot of molecules in a particular pose, it means it's a state of low energy. If you see only a few, it's a state of high energy. And so if we have a conformational map with a trough, then we can follow the direction of low energy. So we get essentially, we get a mapping of the energy landscape by using the Boltzmann formula that I show here. Okay. This is the first time that it was realized, was by Fisher et al. In a paper in Nature in 2010. This is the first application in cryo-EM. And later we followed up in a paper in PNAS. So the applications of manifold EM to date are for the ribosome. That was the initial publication in 2014. And then to the ryanodine receptor that was published in 2020, we can really see the process of activation and gating. And then also the SARS-CoV-2 spike protein was in publication in 2021, where we see the motion of the RBD from close to the open state, which is important for the lodging of the spike protein to the host receptor. So the ryanodine receptor, especially the RYR2, is implicated in many heart diseases. And the person who is studying these is Andrew Marks, who is the chair of the Department of Physiology at Columbia University Medical Center. And so we had a collaboration with two other groups at Columbia, and we were able to get the atomic structure of the ryanodine receptor. The important thing is that the X-ray groups working at Columbia worked for several years, more than 5 years, in order to get a highly diffracting structure, a crystal, and we're not able to get beyond 7 angstrom. And when we got involved, we got into the 3 angstrom range within months. Okay, so here here's the energy landscape produced by this approach. In the case of no ligand, so it's the ryanodine receptor without any of the ligands. The ligands are coffeine, ATP, and calcium. So we get an energy landscape by taking a very large number of molecules, I think 600,000, and analyzing them with manifold EM. We got this energy landscape. And then with ligands, we get this other. How do we interpret this? Well, we can find out the transition probability between the two. And we find 2 hotspots, one here and one there. Let's see. Okay, so from that we can infer that the from the closed state of the receptor, molecules have to travel forward in energy in order to reach this here, and this is the only way how they can, with high probability, transition to the state with ligands. And from that point, there's another downhill a pathway to the finish point. And here we have, you see what is happening. So here, this is now the trajectory that we infer from this. The colors are simply show the distances that the different molecules travel in the process from one to the other state. And this now is in sharp contrast with what you would get from morphing. Morphing is very often done because people don't know the intermediates, and so they do simply linear interpolation, and it's dead wrong because these are completely invented trajectories. Okay, which has no reality. So morphing is a very uh, very dangerous thing. Here's the linear interpolation. So I'm going back to the other one. So you see it's entirely different from what we had before. So I have to go back in order to go. All right. And now a linear interpolation again. Okay. Uh, hmm. So uh, you essentially we're talking about the breathing of molecules and allostery. Small drug binding at target site can be facilitated by allosteric effect of a ligand that is far away. cryo-EM can help in discovering conformational spectrum of a molecule. I think this is just a misplaced slide. I'm sorry. The future, I think, might be a grand synthesis between these two methods. And we have hardly scratched on this. We time-resolved cryo-EM gives us data for a reaction system that evolves over time. And the sightings from molecular ensembles from time-resolved experiments can be linked via the Kramer equation, and specifically the Planck-Fokker equation, in a grand synthesis involving theory and experiment of reaction kinetics. The data can be augmented with data from single molecule red and HDX. Since we have a vastly overdetermined system of equation, albeit with very large amount of noise, we get the rate constants of everything, of every single reaction, backwards and forwards, for the different intermediates. And so in conclusion, single particle cryo-EM can be augmented with two new powerful tools to capture dynamic features of biomolecules: time-resolved cryo-EM of non-equilibrium systems and recovery of the continuum of states in a system at equilibrium. And the time-resolved cryo-EM has been proven to yield novel results on the structure's reaction intermediates as well as reaction kinetics. And the results obtained by manifold EM demonstrate the ability to obtain occupancy maps and energy landscapes by machine learning, paving the way toward predictions of functional trajectories. Okay, thank you very much.
Thank you, Joachim. That was incredible. The, the power of the time resolution and the ability to see atoms moving around at the rate that they actually do is just an incredible extrapolation of the method of cryo-EM. And what did the other people think of this? And would they like to ask any questions? Let's turn over to the audience. Put up your hands and ask. Jonathan.
Thank you. An amazing lecture. So I'm really interested in your thoughts on the manifolds EM methodology. And I'm specifically wondering whether you think that all possible states are accessible through that methodology, or whether if you think about a continuum of states between ground state, transition states, and finished states, for example, in enzymology, does the freezing out process allow you to access all possible states, or do you select out in the freezing process specific, more stable intermediates, leaving you having to interpolate between them?
Well, you mentioned uh, you mentioned transition state. Transition states are occupied only in a very short time, which means that the data collection would be exceedingly difficult to get any measurable quantity of these. So there's transition states are really inaccessible with this kind of method. You would have to get a data collection of millions and millions in order to get the right kinds of amounts.
But if you could acquire millions and millions, would you still expect to see that transition state, or would you still be actually selecting out millions of things that were a bit more stable than the transition state because of the freezing process?
Yeah, the freezing process itself has not been very well studied. There are only theories about this because there is an appreciable time that it takes to equilibrate to the when the molecule is frozen, and there is probably a redistribution of states, and it might very well be that the transition state really disappears in this process.
Can I ask another kind of related question? So how long then before you think that X-ray free electron lasers with kind of femtosecond pulsars, how long before they can reach the resolution that you're getting with cryo-EM?
Oh, that I really don't know. Um, I really have no idea. I mean, yeah, they might never get there. I think there has been a lot of publicity about this, but in terms of investment of money and so forth, I think cryo-EM has been proven to be much more cheap and and productive than any of these methods.
Oh, thank you.
Any further questions? How are we going for time over this week? Click online. Are there any questions online? Just I can't see that.
We are the people online.
Where are people online? Are there any online questions? Okay. No online questions.
Yeah, but what about the other speakers?
These questions, right? Take 15.
Thank you. Is that audible? Thank you. That was amazing. So what has been done in terms of visualizing biological polymers, for instance, DNA, RNA, and their interaction with proteins?
The interactions with RNA or with DNA?
Yes, between enzymes, let's say DNA polymerase and enzymes that bind to DNA, enzymes that bind to RNA, these polymers.
We are in a collaboration with a group at Rockefeller to look at RNA polymerase with a short piece of DNA, so that a minimal piece of DNA. I cannot report anything yet from that, but this is going to be a very promising application of this technology.
Thank you.
there's one comment from Giselle Liver, which was our co-author on the angiotensin converting enzyme. She would have loved to have had access to this technology when we worked on the angiotensin converting enzyme, which Ed is going to talk about.
Yeah, but it's not too late. I mean, there can be a follow-up, all right? Yeah.
It's better for them. Okay.